Hi, I'm Phelan. Let's learn some biology. I want to talk today about how we can use scientific thinking to inform our decision making so that it's more efficient and more effective. You'll recall that we have talked about how the scientific thinking process is not linear, but it's still helpful to think about it as a process where we make observations about the world, we formulate hypotheses about these, and these are our proposed explanations for them. Then we make predictions based on those hypotheses so that in novel situations, here's what we think will happen. We carry out experiments so that we can test them to see if they actually are supported or not supported. And we draw conclusions and we modify the hypotheses, and the whole thing goes round and round. Today I want to get a little less abstract though and talk about really important decision making, like decision making about making cookies. I love chocolate chip cookies, and many years ago I was at a supermarket. I decided, hey, I think I want to make chocolate chip cookies tonight. I was with a friend. I told her, I said, hey, do we have the ingredients for chocolate chip cookies? She said, I think we might need vanilla. So we go down to the vanilla aisle, and I'm looking at the different vanillas, and I grab one, I look, it's very expensive, $30 or something. And then I see next to one, I see a huge container of it, and it's only about $1.50. And I grab that one. And my friend says, no, you can't use that one. I said, what do you mean? She said, that's artificial vanilla. And I said, well, what's the difference? I open it up, I smell it. I said, it smells the same. She said, no, you have to use real vanilla. And I said, why? And she said, everybody knows. I'm like, really? That's your explanation for why we have to use it? And she said, it just tastes better. Everyone knows that. And I said, well, that's not very good evidence that it tastes better. So I grab both of them. I say, all right, let's do an experiment. Let's figure it out. I buy real vanilla and I buy artificial vanilla. I go home. We make a batch of cookies. I have two separate batches, same ingredients, but one of them has all the same ingredients. But with the real vanilla, one has the artificial vanilla. Put them on cookie sheets, swap them halfway through. I do everything so that they're treated equally. And then when I'm done, I blindfold her, I feed her cookies, and I say, all right, rate this cookie. She gives me a rating. All right, rate this cookie. She gives it a rating, and I keep doing this, and we rate a lot of cookies right out of the oven, an hour later, several hours later, and it's clear. She's like, all right, I can't tell them apart. And all the ratings were the same. They tasted the same. So I said, great, I'm going to save us a lot of money over the course of our lives, right? Buying cheaper artificial vanilla. And she said, yeah, but I'd still use real vanilla. I'm like, what do you mean? I said, let's do it again. Let's get even more controlled. So did the same thing, made an entire 2 new batches and rated every aspect and alternated exactly. We did it, and she couldn't tell them apart. She said, yeah, but I said, all right, from now on, every time I make chocolate chip cookies, I'm gonna make 2 batches. I'm gonna make a real vanilla, artificial vanilla. And I did it a couple more times, and then we were having a party, and I overhear, the other side of the apartment, I hear her saying to someone, did you know that you can't tell the difference between real and artificial vanilla? And I thought, I have caused someone to change what they think about the world. They had a hypothesis, now they changed it. And I told someone this story, and he showed me this article he came across. It was a magazine, a website, Cooks Illustrated. And in it, they did a scientific test of all sorts of cooking hypotheses, and one of them was about artificial versus real vanilla extract. And it was funny because they said, we were so surprised by the results of this test, we had to do it again. Same experience that I had, and what they found was that actual professional chefs could not tell apart the cookies that were made with real vanilla versus artificial vanilla extract. So you do this and you can find out. It can change your mind. I told this to my brother. He sent me a picture of this, of all the different vanillas. He claims he can detect taste differences in all of them. I haven't done that experiment yet, but I feel like we've learned an important lesson, or I've learned an important lesson. Now, the thing that I did there is what we call a critical experiment. A critical experiment is where you put your hypothesis to the test. You design a way of collecting observations so that when you get your results, it's going to either support a hypothesis or support a different one. And you can't hold both of them at the end. So when you're designing what we'd call a critical experiment, you think, all right, well, what information could I get that would cause me to actually say, all right, I got to give up that idea. And then you try to collect that information. And if you think about just all the things in the world that you might think about, there are a lot of hypotheses that you can come up with. So, does taking a vitamin supplement reduce the likelihood that you will get sick, or does it reduce the duration of the illness? Think about it. We could figure this out now. There are experiments that we could do. In fact, people have done them. We know the answer. Or antibacterial hand soaps. You go to the supermarket, you see them, you're like, whoa, well, yeah, I got to get those. I want to, kill the bacteria. Do they actually cause you to get sick less or to stay sick longer? We could figure this out with a critical experiment. Think about how you would do it. Dividing people into groups, giving them one versus the other, not telling them which one, having them keep track of some sort of illness journal for a year, swap the groups, and now they're in the other group, so each person's their own control. I'll give you a little tip-off on this one, that the antibacterial agent that's in most antibacterial hand soaps, triclosan, guess how long it takes to have its effect? 2 to 3 minutes. Now, how long do you wash your hands for? They've done hidden camera experiments in bathrooms. It's about 20 seconds. So even if it did matter, which it doesn't, you're not washing your hands long enough. They're not exposed to it long enough to be killed by the triclosan. So that's a critical experiment. We have talked last time in the previous video, I talked about, could you figure out, do cell phones cause brain tumors? How could we figure this out? There are some critical experiments that you could design to figure this out. Here's one. You might think you don't know anything about this. Let's say we wanted to know, is maze running behavior in mice genetic? Is there a genetic component to this? Think about how we might be able to do this. We might have this idea in the first place because you notice that there's a lot of variation among mice in how well they learn to run a maze. People have made these observations. So if our idea was, hey, I think maybe maze running behavior is influenced by genes, what could we do? You think about, all right, well, I don't know anything about mice, you might think, but you could still come up with some experiments. So for instance, I could say, what if you had a big population of mice, you train them on a maze, you see how long it takes them, then you train them an hour later or the next day or the next week, and you map out for each animal how quickly does it master the maze. And so for each animal, you'd have some index of how good are they at learning a maze. Not the first time they run it because they've never seen it, but how much do they remember? How much do they improve? Then what if you took the top 20% of the maze runners and you bred them with each other and now produced a bunch of offspring from them? And simultaneously, you take the 20% that are the worst at running mazes and you breed them with each other, get a bunch of offspring. Then from each of those groups, you say, all right, now we're going to train all of these animals on the mazes, and we're going to rank all of them on how good are they at learning maze running behavior. And then you could take the top 20% of them. So the top 20% of the individuals whose parents were already in the top 20%, and then from the other group, maybe take the bottom 20% of the bottom 20%. And you could do this over and over and over again. Let's say you did this for 4 or 5 or 6 generations, and then at the end, you take your populations and you test them and you say, all right, how quickly do these animals, they're new animals, they've just been born, how quickly do they learn how to run a maze? If there's no genetic component to maze running behavior, they should all just be the same as they always were. But some researchers in the 1950s did this, and look at what they found. They put the animals in, they did this, they measured how many errors did they make in the maze, average number of errors, and look, over the first, second, third, fourth, fifth, sixth generation, what we see is the maze dull animals, they call them. They progressively were making, on average, more and more and more errors. The ones that were maze bright were making fewer and fewer and fewer and fewer until at the end, these maze bright, they could learn them so quickly they couldn't even build mazes that were complex enough for them to be challenged by. So there's a pretty good critical experiment. Think about this. Could you do this for humans? They actually have mazes made out of hedges. And at first they seem fun, you walk around. But as you go further and further back into the maze, they let the hedges get higher and higher and higher until pretty soon you're way in the back. Now you literally are just a rat in a maze. And they make you, when you do these, it's like entertainment. When they make you do these, they give you a white flag so that if you're in the back and you panic, you can't get out, you hold up your white flag and they chopper someone in to airlift you out and put you in the maze dull group. So this would be an important take-home message. If you're going to figure out what you should think, if you're going to change your mind, you want to design a critical experiment to help you do that so that the results are going to unambiguously tell you this interpretation or this one is consistent with the observations. Once you've done that, you still want to figure out, because doing experiments, collecting observations is time consuming and expensive. So you want to do it in the best possible way so that you are going to get maximally useful information. One of the ways is you want to randomize individual subjects to your different groups so that you get all the subjects and then you randomly put them into the group that is going to get the vitamins, or those that are going to get the placebo, or the group that are going to get some muscle building compound versus those that aren't. You want to randomly put them rather than letting them select it. That way there's no underlying difference between the groups. You want to control your study as well, and control is at the core. When you are trying to figure out the impact of a particular intervention, you want to make sure that some individuals get the intervention, some don't. But other than that, everything is consistent between them. So trying to control an experiment, that's at the heart of designing a good critical experiment. A topic that I'm extremely interested in is sleep and being tired. We see it in animals and humans. There was a claim made, and I'm sure you've heard this before. They said this. They published this study saying that lack of sleep increases your risk of getting sick. And here's what it said. Facts: as cold season approaches, many Americans stock up on vitamin C, blah, blah, blah. Studies have demonstrated, in a recent study for the Archives of Internal Medicine, scientists followed 153 men and women for 2 weeks, keeping track of their quality and duration of sleep. The people did this by filling out little sleep diaries. Then, during a 5-day period, they quarantined the subjects and exposed them to cold viruses. They then made a comparison between those who, prior to quarantining them, those who slept a lot and those who didn't. They said this: those who had slept an average of fewer than 7 hours a night, it turned out, were 3 times as likely to get sick as those who averaged at least 8 hours. So you see what they did? They've figured out a bunch of people, some of them not sleeping very much, some of them sleeping a lot. Now they bring them all in and they expose them to the virus that causes colds, and they see who gets sick more, who gets sick less. And they find that those who don't get as much sleep are significantly more likely to get sick. Now, think about this. Is that a great study? I mean, it's published, it was interesting, but do you think that the group of people who were getting a lot of sleep and the people who weren't getting any sleep at all otherwise were identical? I don't. Right? What could be different? Well, maybe the people who weren't sleeping much, they had multiple jobs. So they were stressed out because they had financial stresses. Or maybe they didn't have enough money and needed multiple jobs, or they had to work more hours. And because of that, they didn't have proper health care. So maybe they hadn't seen their doctors as much. You could come up with a bunch of different reasons why there may have been underlying differences between these two groups. And because of that, then you're not sure you're attributing this difference in how likely they are to get sick or not get sick to their amount of sleep, but maybe it's to some underlying thing. Did the people who are getting less sleep all have more kids? And therefore, maybe their kids are at daycare and they're bringing home more viruses from their exposure there? We don't know. So think about how you design that an experiment to test that exact same question, but that would be maybe a little better. So when you're trying to control an experiment, you want to make sure that the groups are exactly the same. So maybe you recruit 1,000 people and you randomly put them in the low sleep or the high sleep group. And you enforce this maybe by quarantining them somewhere, and one group gets really nice, cozy, quiet place where they can sleep. The other group, you're blasting alarms at a certain time so that you control how much sleep they get. Then expose them to the virus, see who gets sick more or less. Then you could swap them, put them in different groups. So now you have each person under both conditions, and you can see whether or not they're more or less likely to get sick. That would be a better way to control that. So it's not that it's a useless study when you haven't controlled it perfectly. Sometimes that's the best you can do at first, and then you progressively do a better or more well-controlled study. There's an additional type of control that people use called double blind or blind. Blind just says that the person who's receiving a treatment, they don't know whether they're getting the actual manipulation, the thing that you think is going to have some beneficial effect, or a placebo. So that's blind. Double blind says even the person administering it doesn't know which group they're in. That way they don't convey anything subconsciously about, oh, I think you're going to feel better this week, or, or something like that. Could be nonverbal even, but that can have an effect. So if it's double blind, the medicine just is labeled with a number, and someone in the back lab knows what it is, but the person administering it doesn't. So double blind study is a nice way of controlling it. I did an experiment years ago. I did it with my wife, Julia, and we were interested in symmetry. We were looking at left-right symmetry in humans. We wanted to know, were some people more symmetrical than others? And we had a hypothesis we were testing about biracial people maybe being more symmetrical. And so as part of this study, we had to measure people, their left index finger and their right index finger, and we'd see, are they the same? How much do they differ? Then their ring finger, then their wrist, then their ear length, and all these different measures. And we would compare the left and right side, and you'd see what was the difference between them, divided by the average of those, and you'd figure out, oh, there's this much deviation between the two sides, and you'd sum it across all the different things. Well, sometimes it was because it was hard to figure out where exactly to put the calipers. You'd be like, whoops, oh no, I think I put it in the wrong place. And what Julia did was she said, hey, we have this idea about which group is going to be more symmetrical than another, and so we shouldn't be deciding. Sometimes we're going to redo it because maybe we didn't get it in the right place. And that was a smart idea, that we might have been subconsciously sort of deciding when we should remeasure versus other times not. So she came up with this idea in this case that's sort of a version of double blind, which is that she covered up the indicator of what the reading was with tape. And then she had a little cable that went to a computer, and you would just push a button. So if you thought you hadn't done it right, it wasn't because you saw the number. You just thought you hadn't done it right, and you would redo it. But now it wasn't based on seeing what the data were. So that's a way that you can increase the control of your study. Now, here's a question for you. Let's say you've got some drug that you think is going to be effective in fighting the AIDS virus. Now, what's going to be a better way to test this? To do it in the lab or to do it in a natural setting? People debate this a lot. What study would be better? A lot of people say, oh, well, lab studies are the best, obviously, because you can have lots of samples, and you can control the temperature, and you can control the light, and you can control the humidity and nutrients and all this. So any difference between two groups you can attribute just to the one thing that differs between them. And there's a lot of validity to that. But then at the same time, someone will say, yeah, but we're not really interested in, does this drug kill the AIDS virus in lab cultures? We're actually interested in humans. So then you think, all right, well, we got to do a real world study. And this applies not just for pharmaceutical tests. This could be any test, a test of behavior in wild animals or anything. And in real world situations now, you have a much more realistic scenario. that you have the organisms in their native habitat. But in this example of the AIDS virus, all of a sudden now you're working with people and maybe they don't take the drug at exactly the same time each day. Or maybe their nutrition is better in one group than the other, or it varies a lot. And your ability to control stuff can be limited. So you've got this trade-off that you're doing where the more you control it, the greater your confidence in the results you have. So that's good. And that kind of nudges you towards the nice lab environment. But typically as you get better and better and better controlled experiments, they become less and less and less like the situation that you're actually interested in understanding better. So there are trade-offs. You do both. Sometimes you do them at the same time. Sometimes you do them in sequence. Sometimes you work with other people. But it's not like there is a better way to do it. Lab studies are better, or field studies are better. One last thing I want to throw out there in terms of you trying to decide what do you believe? How do you make decisions? Is you have to be skeptical. Skeptical doesn't mean you don't believe anything. Skeptical means that you ask all the time. How do you know that's true? And here's something. The London Times had this article where they said 1 in 7 fathers not the real parent. Now that's a super provocative headline, especially if you've got kids. You're thinking, hang on, 1 in 7? That seems high to me. But, sucks me in. I want to read it. And what do they say? Well, they say Cellmark Labs gets more than 10,000 requests a year for paternity testing. 1 in 5 of these hasn't been ordered by a court or child support agency. And of these, the ones that hadn't been ordered, in 1 case in 7, the presumed father turns out to be the wrong man. So that's what they said. Now, how do you feel about that? You've just read some data, some conclusion that someone made. How do you feel about that? Well, think about these people, the 1 in 5 that wasn't ordered by a court. These are still people who said, I think I'm going to go online and do a search for paternity testing agencies. And, here's Cellmark Labs. I'm going to go to them. Do you think that that's representative of the entire world? Maybe not. So maybe if they say 1 in 7 fathers of people who have sought out a paternity testing situation, maybe 1 in 7 of those fathers is not the real parent. Okay, I'm fine with that. That's not what their headline said, though. So you need to look at things and think, all right, well, did they control, is that actually representative, the population they looked at, with the population that they are making some assertion about? You got to do this all the time, and hopefully you'll find out that it is a reasonable conclusion to make, but not always. So that's scientific thinking and decision making.